Okay, the first chapter that we're going to talk about is DNA replication. It covers a very important step that happens in the S phase of the cell cycle. Okay? So we're talking about DNA replication. Okay, so this is the what are we talking about? Okay, the answer to the question when does this happen? We said S phase. Very good. Of the cell cycle. Good. No, so what it is your DNA being copied identical. So whatever the DNA is, you're gonna now have 2 copies of it. Outside of random mutations, and we're not gonna talk about mutations right now. That's later. So if we start by just writing a random sequence, your DNA would look like this: A, G, C, and T. It's double-stranded because that's what we said in chapter 5. 5 comes back to haunt you right now. Chapter 5 comes back to haunt you right now. A, G, C, and T are the 4 nucleotides. And then it's complementary. A is complementary to T, G is complementary to C, C is complementary to T is complementary to A. Got it. These are covalent bonds. They're called phosphodiester. You got it. And this is 5 prime. That means this is 3 prime. And if this is 5 prime, this will be 3 prime. If this is 3 prime, this will be 5 prime because it's anti-parallel. These are phosphodiester bonds. They're covalent bonds. But what are the bonds holding the strands together? Hydrogen. Very good. So these are hydrogen bonds. I hope you realize that none of this is new. Do you agree?
Yes.
Good. The nucleotides are the A's and T's. Those are the monomers of DNA. You attach them together through dehydration synthesis to make the strand, to make the polymer. But once you have 2 strands, they come together because DNA is double-stranded. At the 5 prime end, there's a free phosphate. At the 3 prime end, it's a hydroxyl group. Well, same thing here. That's the phosphate, that's the hydroxyl. None of this is new. Okay, the new stuff begins here. When we go copy it, what happens? Well, when we go copy it, both strands will separate. So it's an enzyme machinery, and that's a tough part. But I'm going to give you the gist of the overall event. The overall event, what's going to happen in the S phase is your DNA is going to separate, and you're going to copy this strand, copy the other strand. Done. It's the quickest way to copy DNA. Watch this. If my elbow is the 5 prime phosphate and the tip of my finger is the 3 prime hydroxyl, this is the other strand because DNA is parallel, but it is anti-parallel. Very good. Here's how I'm going to copy it. Separate the strands. Copy this one. I'm done. Once I'm done, I'll have 2 copies. It's the easiest way to do it. So let me show you how that would be done. Now it's an involved process, and we're going to talk about it. This is now DNA replication. Big picture. I will separate the strands. So A, G, C, and T. One of the strands will be separated from the other one. This is the 5 prime phosphate. This is the 3 prime hydroxyl. This is the 3 prime hydroxyl, and this is the 5 prime phosphate. I separated them. And how do I separate them? All I have to do is break the hydrogen bonds. Can you see that? And then copy, done. Copy would be like this. For every A, there's a. For every G, there's a.
C.
Keep going.
G.
Keep going.
A.
Good. Do this one.
C. G. A.
Good. And if this is 5 prime, what would this be?
3 prime.
If this is 3 prime, what would this be?
5 prime.
Good. Same thing with this one. If this is 5 prime, this is This is? Got it. I'm done.
That's it.
Well, the big picture. When we talk about the enzymes involved, you realize, oh, he just summarized it. This is just a summary of the event. I wish chapter 15 was like that. It would be done in a second. But it's not. I will tell you what happens here. This is the result of DNA replication. You have one copy, now you have two. Do you see that? But there's something interesting about the two copies. You may not see that right now, but as soon as I say it, you'll realize it. The interesting thing is this. Half of the copy is the original strand that I drew in black. The other half, I just made for you in red color. And the other copy has half old, half new. No. It's a process called semi-conservative replication. What did you say, 50%, right?
50%
No.
original.
Yeah, 50% original, 50% parental. But we call that semi-conservative replication. The people that discovered this were up against 3 possible theories. And your book talks about it. And this is the one that won. I don't want to even talk about the other ones because they're not valid. Conservative, dispersive, it doesn't do that. All DNA replicates semi-conservatively, which is a fancy way of saying this. DNA replicates this way. Watch. Separate the strands. Copy. You're done. And when you do that, one of the copies will have the strand from the original and the new one. The other copy will have the other original and a new one. That's called semi-conservative replication. This is in contrast to if the two new ones, the new ones were made up of these two and the original ones. You see what I'm talking about? If the cell decided, I'm going to put the two new ones together, I'm going to keep the two old ones together. That would be conservative replication. That does not happen. That's why I don't want to talk about it. Conservative would be putting these two together and the two original ones together. How is the cell going to know that to do that? This strand is no different than that one, is no different than that one. Just because it just made it, it doesn't care about that. Dispersive is even worse. So, no, I don't do that. All DNA replication, I don't care who you're talking about, is made this way. Simple. Separating of the two strands. Copy this one, copy that one. You're done. The end product will be half parental, half new. Half parental, half new. And that's called semi-conservative replication. So we're going to put that as one characteristic of DNA replication. It is semi-conservative in the sense of replication of DNA is semi-conservative. DNA itself is antiparallel. DNA itself is made up of nucleotides. DNA itself, A is complementary to T, G is complementary to C. That's the characteristics of DNA. It is a helix molecule. But when we go to the replication of DNA, the copying of the DNA, we start throwing in new terms here. It is semi-conservative. That means the two copies are half old, half new. Half old, half new. Are we okay so far? Okay. You have it on video. I'm going to erase this, and I'm going to keep going. I'm going to see if I can build on the characteristic of DNA replication here. Okay. I'm going to explain to you this arrow. When Lordson said, is that it? No, watch this one now. Look at this arrow. It's deceiving. Here we go. The fun begins now. This was just the trailer. Okay? This is the real movie now. All right, I'm going to write I'm going to do just a line as the sequence. Here's how your DNA begins. It is double-stranded. It's got hydrogen bonds all the way through, holding the strands together. That's your DNA. The first enzyme that comes in play is called topoisomerase. Topoisomerase is the first enzyme that comes in play. Topoisomerase will recognize a sequence on your DNA called the origin of replication. There's a lot of words coming your way. It's the language of this stuff. Let's say the origin of replication is right here. It is abbreviated ORI, which stands for origin of replication. It is sequence dependent, meaning there's a sequence for that. It says, oh, that's my origin of replication. Based on the sequence, it knows that. Topoisomerase will bind to the origin of replication, remove the hydrogen bonds. It will do this, remove the hydrogen bonds. And when it does, the DNA bubbles out. So it will look like this, if I can draw it. Not very good at that. Not a good artist here. It will look like that. And you'll still have the hydrogen bonds. That's fine. But you won't have the hydrogen bonds here in the origin of replication. Why? Because topoisomerase broke those. Now, to sidetrack a little bit, in eukaryotic cells, our DNA is linear and long. Linear and long in eukaryotic cells. In prokaryotic cells like bacteria, it's circular and smaller. For eukaryotic cells, they can afford to have a single origin of replication because it's circular and much smaller in size than our DNA. Our DNA is long and linear. We can't afford a single origin of replication. See, the word origin of replication means that's where the DNA replication will begin. You can have one start point in bacterial DNA because it's small and circular. But in linear DNA, like our DNA, where it's long, I can't do that. If I have a single origin of replication in our DNA, good luck with that one. Because this is you waiting for your DNA to be copied. You're dead. You can't afford that. You want your DNA copied right now. I'm not going to wait for this. It's a long piece of DNA. The only answer to this is, watch this, multiple origin of replications. That's the main difference between us and bacteria. Our DNA is long and linear. Their DNA is shorter and circular. They can afford to have a single origin of replication. But us, that would take too long to copy. So we have multiple origin of replications, and the copying is going to be happening here, here, here for efficiency. I need my DNA copied right now, and I'm not going to wait for it. So the only solution in eukaryotic cells is we have multiple origin of replications on the chromosome. Bacteria has a single one. There's a reason behind that, because of number 2 on the list. Number 2 on the list is DNA replication is bidirectional. Meaning that when the DNA gets copied, it goes in both directions at the same time. Efficiency. Always think about your cell being extremely resourceful, extremely efficient in what it does. And the best way is, hey, might as well goes both ways, bidirectional. Well, it's the same thing here. Your DNA replication is bidirectional at each origin of replication too, for efficiency, for speed. So the only way to guarantee that in eukaryotic cells is to have multiple origin of replications. And that's a good question on the exam. What's the difference between DNA replication in bacteria versus eukaryotic cells, when it's not because it's semi-conservative. All DNA replication is semi-conservative. It's not that one is bidirectional, one isn't. All DNA replication is bidirectional. It has to do with the number of origin of replications. Bacterial cells have one origin of replication in their chromosome because it's small and circular, whereas ours is long and linear. So we have to have multiple origin of replications for efficiency, for speed. Okay, that's just a side note. You'll see that in your PowerPoint slides. Obviously, I'm not going to use PowerPoint slides today, for the most part. Now that I have this opened like this, their terminology is here. This part right here creates like a V-shaped structure. Watch this. Look up here, please. It looks like this. Well, then there's another one on that side too. So if you think about it, it's like a mirror. Pretend as if there's a mirror in between here. And realize that whatever is happening on this side is happening on that side, because DNA replication is bidirectional. There you go. Whatever happens this way is happening this way. The symmetry point is the imaginary line down the origin of replication. So this area right here creates like a V-shaped structure, and this area here also, that's called the replication fork. This region is called the replication fork. So if you look at it, for every origin of replication, there's two replication forks. One on this side, one on that side, because DNA replication is bidirectional. We okay so far? Yeah. All the events that are happening here, or they're gonna happen here, will also happen here. It's just that this is going this way, that's going that way. Okay, next enzyme in line is another topoisomerase. It has the same name, it's just will have a different number. Don't worry about those numbers. One of the topoisomerase will bind to the origin of replication and cause the bubbling to form the replication forks. See how we're changing it into words now, into a story. Once the replication fork is formed, another topoisomerase will come in. Now watch this. Will bind ahead of the fork. So I'm going to redraw it as the best I can. And again, here are the hydrogen bonds. Those are the same. Hydrogen bonds. Origin of replication. The imaginary mirror splitting it in half. Okay, this topoisomerase, I'm going to draw in green, is going to sit right here. And I remember him saying, whatever happens here, happens here. Because it's bidirectional. What does this topoisomerase do? It will do this. Untangle the DNA ahead of the replication fork. It's sort of like mom saying to you, you gotta untie your shoe before you take your shoes off. You gotta untangle the DNA before you copy it. Who's the untangler? Topoisomerase. Next enzyme in line is gonna be helicase. This enzyme will sit right here, and it will break the hydrogen bonds, making the bubble bigger. So I'm going to try to draw that. The bubble got slightly bigger. Again, you still have the hydrogen bonds downstream from there. That's fine. Those are hydrogen bonds. You still have the origin of replication marking, and then the line of symmetry. You still have the topoisomerase untangling the DNA ahead of the fork. Now you have, try to do different colors here. Now you're going to have helicase. So this is topoisomerase, and this one is going to be helicase. Of course, whatever happens here happens here. Now, helicase unzips the DNA by breaking down the hydrogen bonds. See, topoisomerase untangles the DNA ahead of the fork. Helicase unzips the DNA by breaking the hydrogen bonds between the A and the T and the G and the C. So if you look up here on the board here, you guys, you'll see me doing it. My finger is like helicase. Watch this. Hydrogen bond, break, 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 break. That's what it does. So it's changing. The double-stranded DNA is becoming single-stranded. See, it's double-stranded wherever you see the hydrogen bonds. So this region here is double-stranded DNA because the two strands are hydrogen bonding with each other. But this area here is single-stranded DNA because it's no longer hydrogen bonding with the other strand. And that's the job of helicase. You haven't copied anything yet. You're just preparing it. Helicase unzips the DNA to make it single-stranded DNA. How? Simple. Break the hydrogen bonds. You're not going to go boiling anything here. You're not going to add heat. This is an enzyme doing it. Because I know we can use heat to break hydrogen bonds, but you won't be able to do that with this one. You'd need excessive heat, and you'd die from that heat. So it's going to be enzymes doing this. All right, then what happens? What happens comes in, and another protein comes in. This is a protein, it's not an enzyme, called single-stranded binding protein. That's bad. And it lives up to its name. It only binds to single-stranded DNA. That's it. If you give it double-stranded DNA, it says, I don't know what to do with that. I'm not going to bind to it. I'm a single-stranded DNA binding protein. You give me single-stranded DNA, I'll bind to it. Double-stranded DNA, I don't bind to that. So let me show you how that would be in this case. It would look like this. Here's the bubble. The other strand with this bubble, that's a bad one. Let me try that. The hydrogen bonds. The green for the origin of replication. The symmetry line. And what color did we use? We used green for the topoisomerase. We used this one for the helicase. And here comes the single-stranded binding proteins. The single-stranded binding proteins will do this. Wherever there's single-stranded DNA, I'm going to bind to it. This is the origin of replication. These are the single-stranded binding proteins. So what they do is they prevent the A and the T from rehydrogen bonding. They prevent the G and the C from rehydrogen bonding, meaning they prevent the DNA from rezipping. See? Helicase unzips your DNA to get it ready for DNA replication. If it rezips, then you won't be able to copy it. So something has to keep the DNA unzipped, and that's the job of these guys, single-stranded binding protein. You haven't copied anything yet. You're just preparing it so far. All right, next one that comes in play is an important one. The next one that comes in play is an enzyme called primase. This enzyme makes what is called an RNA primer, which is a fancy way of saying a small piece of RNA. How long? Whatever, 20 bases, let's say. Small piece of RNA. Okay, based on what sequence? Based on the template sequence of your DNA. So let me redraw it, and you'll get an idea what I'm talking about. I'm going to redraw it again, so you can have pictures of every event, hopefully, by the end. That's a bad one. Better. All right, origin of replication. The single-stranded binding protein. Change the color of the origin of replications to red. Okay, I'm gonna have the double-stranded DNA on this side. Ooh, that got a mixture of things. Okay, I'm gonna have the topoisomerase. The helicase is unzipping the DNA by breaking down the hydrogen bonds. And here comes the primase. Remember that symmetry, that line of symmetry? This becomes important now, watch this. Whatever is happening on this side is happening on this side, but there's a catch to it. It's an upside down image. If you ever go to the circus or whatever, they have these mirrors where you stand in front of the mirror, you see a reflection of yourself upside down. So whatever is happening up here is happening down here. Whatever is happening down here is happening up here. So primase is going to come in and lay down a primer going this way. That means another primase is going to come in and lay down a primer going this way. So there's an enzyme called primase that's going to do that. Primase is going to do that. And it's laying down a small piece of, catch it, RNA. Some students stop me here and say, wait a minute, I thought we're making DNA. Yeah, but that's how it starts. And now here we go. If whatever is happening here, another primer is going to be laid down right here. Watch this. Primase is going to kind of come over here and make a primer this way. That means primase is going to come over here and make a primer this way. By the way, of course, there's single-stranded binding proteins here too. So at the beginning, we're talking about 4 primers. They're all RNA. 2 here and 2 here. Now comes in play the third thing about DNA replication. And the third thing is this. When you're copying DNA, you always, listen carefully to this one, you always make the new DNA in this direction. We talked about this before a long time ago. The new DNA is always made 5' to 3'. So we're going to start making DNA over here. At the end of the primer, look up on the screen please, so you can follow it. At the end of the primer, another enzyme is going to come in and start making DNA. If I orient this molecule, this is the 5' end, this is the 3' end. So if I start making DNA this way, then I'm going 5' to 3'. You make the new DNA 5 prime to 3 prime. All the time. Same thing with this one. This is 5 prime, this is 3 prime. The new DNA is going to go this way. Same thing with these two. This is 5 prime, 3 prime. The new DNA is going to go this way. And same thing with this one. I'll make this a little shorter. The new DNA is going this way. New DNA is always made 5 prime to 3 prime. Here's the confusing part. To make this DNA 5 prime to 3 prime, listen, I have to read the template 3 prime to 5 prime. The template orientation is this way. So this 3 prime is the same as having 3 prime on this end here. So I'm actually reading the template 3 prime to 5 prime to make the DNA 5 prime to 3 prime. It has to be done that way. Well, it's the same thing for this one. I have to read the template 3 prime to 5 prime to make the DNA 5 prime to 3 prime. So there's another layer of concern here because of this rule. The rule that cannot be broken is this one. I must make the new DNA 5 prime to 3 prime. That means I must read the template 3 prime to 5 prime. Well, that's easy for this one. Because all I have to do is start making the DNA and just keep going until I finish. So watch this. I'm going to do a fast forward of the event here. If I bind here and I start making the DNA, I don't stop until I reach the end. So I use the tail end of this primer to build DNA all the way until I finish it. Who am I? I am the next enzyme, DNA polymerase, doing this. So the next enzyme that comes in play is DNA polymerase 3. That's the guy that's doing that. DNA polymerase 3 will bind at the tail end of the primer and start making DNA. On this one and this one, it can afford to go all the way through until it reaches the end. This strand is called the leading strand. The characteristic about this strand, and the same with this one, that's also the leading strand. There's 2 leading strands, one for each one, going in opposite directions, like we said before. It's made continuously, which means, fancy word for saying it's made in one piece. You'll understand it when we see the next one. This strand is made in one piece, and so is this one. So they're both called the leading strands. This is different than the other one. See, the other one has a dilemma. The dilemma is, I have to follow rule number 3, 5 prime to 3 prime. But the problem with to follow that, I have to read 3 prime to 5 prime. The problem is this. When I was making this one, the DNA was opening as I was copying it. Are we catching that?
Yeah.
As I'm copying it, the DNA is opening so I can read some more and keep copying. I can't do that with the other one. The other one is a problem. I can't copy to make the RNA primer until it opens. So I wait for it to open. I make the primer. Then DNA polymerase 3 comes in and makes the DNA. I wait for it to open. I make the primer. DNA polymerase 3 comes in, makes the DNA.
And you are?
I'm now the second strand. That's called the lagging strand. Watch, you'll hear the whole thing and then give me questions, okay? That's the lagging strand. I am made in pieces. Lagging strand is made discontinuously, which means pieces. The name of the pieces is Okazaki fragments. That's the name of the pieces. So that's an Okazaki fragment. That's an Okazaki fragment. That's an Okazaki fragment. So let's do it again. DNA polymerase, the primates will lay down an RNA primer. Here they are. DNA polymerase 3 will come in, bind to the tip of the RNA primer, and start making DNA all the way through. That one is called the leading strand because as the DNA is opening, it's being copied at the same time. So no interruptions with that one. It's made continuously, and that's the leading strand. And remember, whatever happens here, just an upside down mirror image. The problem is on the lagging strand. Because the DNA has not opened yet. How am I going to make the primer if the DNA is still double-stranded? So I'll make the primer wherever it's single-stranded to start making the DNA. And then I'll wait a second. When it opens up, I'll make the next primer, copy the DNA. And then I'll wait another second. When it opens up, I'll make another primer, make the DNA. That means I'm making that strand in pieces. That's what it means. And it all has to do with the fact that DNA is anti-parallel. And the rule of all DNA must be synthesized 5' to 3'. I mean, your cell is amazing. It figured out a way around all this stuff. Okay, that means I'll make the first one, the leading strand, continuously, one piece. But the bottom one, I have to make in pieces. Problem solved. Again, whatever is happening here is happening here. But it's just upside down. Inversion there, watch. Lay a primer in the single-stranded single-stranded area, make a DNA. And then once this one opens, I'll make another primer. And then DNA polymerase 3 will come in, make DNA. And then once it opens again, I'll make another primer. DNA polymerase 3 will come in and make the DNA. And that means I'm making it in fragments. What's the name of the fragments? Okazaki fragments. Yes, sir. You good? So, we're going to backtrack and look at the need for this primer. DNA polymerase 3 has to have RNA primer for it to do its job. It's a must. It's sort of like me. I have to have a projector and a computer to do my job. Without, I don't work. So RNA primer has to be there for DNA polymerase to do its job. Look what would happen otherwise. Watch this. I am DNA polymerase 3. I have 4 nucleotides, A, G, C, and T. Okay? So I am DNA polymerase 3. I know how to make DNA. I'm going to move this here, sir. I can lay down DNA. I just read the nucleotide and bring in the complementary. So if I read A, which nucleotide am I bringing in? T. If I read G, which one am I bringing in? See, I'm very good at what I'm doing. I'm DNA polymerase 3. But I have a problem. If you don't put the primer there, look what's going to happen. I read A, I bring in T. There's nothing to attach it to. I read G, I bring in C. And it falls away, everything is falling apart, right? There's nothing for me to attach the nucleotide to. I want to work for you. Here's another problem. Without the RNA primer, I don't know where to start. DNA polymerase 3 will come to work and realize, oh, that's a long DNA molecule. I wonder where he wants me to start. Because he didn't mark the start point. So the RNA primer serves 2 jobs. Job number 1, it tells DNA polymerase 3 where to start. That's job number 1. Job number 2, it gives DNA polymerase the attachment point, the hook. Watch this now. Primase comes in, and it first lays down an RNA primer. That's it, RNA primer. So wherever the RNA primer is, that's where DNA polymerase III should start working. It's not going to look over there to start. It's not going to look over there to start because it tells it, oh, I need to start here because that's where the primer is. So wherever the primer is, the primer says, hey, DNA polymerase III, yeah, I want you to start here. Okay, now the primer is here. What the primer is giving DNA polymerase III is the hydroxyl group, that hook, that it can now do this to. A, T. Is it falling? No, because you got the RNA primer. Okay, G, C, C, G, and so forth. It's now building the DNA. Now that's the purple line. Are we catching that? So DNA polymerase III requires the primer to be laid down by primase because the primer does two things for DNA polymerase III. It tells it where to start and provides the first attachment point. So then it can grow from there. In which direction is it growing? 5' to 3', because the cap is the phosphate and this part is the hydroxyl. Then the next step is DNA polymerase III's cousin, DNA polymerase I, comes in and says, I don't need this one anymore. What is that? That's the RNA primer. I don't need this one anymore. And it breaks it down and replaces it with DNA. So the next enzyme that comes in, I'm just going to write it. DNA polymerase I will remove the RNA primer because it already did its job and replace it with DNA. So at the end, all you're going to have is DNA because that was the intention anyways. But if you realize something, the leading strand is going to be in one continuous, is continuous. So we always say that leading strand synthesis is continuous, which means it's made in one piece. But we say lagging strand synthesis is discontinuous, which is a fancy way of saying it's made in fragments. What's the name of the fragments? Okazaki fragments. The concern is at the end, that when you do have DNA made in pieces, how are you going to seal these pieces so that it would look like this? Well, there's another enzyme. The other enzyme is DNA ligase. The last one, thank God. DNA ligase. DNA ligase will come in and seal the gaps. They're just single phosphodiester bonds. And it will make the DNA look like this. A straight strand. So at the end, you will not know the difference between the leading strand and the leading or the lagging strand because they will both look like this, full strand. So we say DNA ligase seals the gaps. Seals the gaps. We're not talking about a gap that looks like this. We're talking about a gap that looks like this. You see? The enzyme goes and binds there and seals the gap. That's it. All it's doing is creating a phosphodiester bond. That's all. Dehydration synthesis, phosphodiester bond. And at the end, you're going to have a complete DNA strand fully sealed. Questions on this? Because that's the process. That's the process of DNA replication and all the enzymes involved. So now, if I ask you to quickly explain the process in, let's say, 3 minutes or so, 2 minutes or so, you would say something like this. Double-stranded DNA will bubble out at the origin of replication by topoisomerase. Another topoisomerase will bind ahead of the fork to untangle the DNA. Helicase will come in and unzip the DNA by breaking down the hydrogen bonds, making the double-stranded DNA become single-stranded DNA. To keep it single-stranded, the single-stranded binding proteins will do that. Single-stranded binding proteins will come and bind to the single-stranded DNA to keep it single-stranded. The primates will come in to lay down the primers. DNA polymerase 3 will now know where to start and will have the attachment point to begin making DNA. On this strand, DNA synthesis is continuous. That's called the leading strand. On this strand, DNA synthesis is discontinuous. That's called the lagging strand. Look at how I remember this one. The leading strand is the one that's going towards the fork. It's going towards the fork. The one that's going away from the fork or towards the origin is the lagging strand. So you can say the leading strand is moving towards the fork or away from the origin. That's the leading strand. The lagging strand is moving away from the fork or towards the origin. It depends on how you look at it. And then, once DNA polymerase 3 makes the DNA polymerase 1 realizes that we don't need the primer anymore. So DNA polymerase 1 will remove the primer, replace it with DNA. As a result, you'll have a gap between the two DNA fragments called Okazaki fragments. To fill the gap, or to seal the gap, DNA ligase comes in and does that. That's the whole process. You need to know the enzymes, the order of their appearance, and what they do. You also need to factor in the importance of RNA polymerase and the fact that DNA polymerase 3 cannot work without this primer. Are we okay with it? All right, hang in there now. We have an issue now. The issue comes in towards the end of the chromosome. I'm going to expand this part right here on the other board, okay? So the issue comes in at the end of the chromosome. What happens? Okay, let me expand on this here. I need your attention, please. You can tell this one is going to be mind-blowing for you. The leading strand is no problem. The leading strand, which is the one coming this way, we're going to be able to reach all the way to the end. So the DNA polymerase 3 will keep extending until it reaches the end. So you're going to get the whole strand copied. That's not where the problem is. No problem making the leading strand. No problem for the leading strand. The problem is with the lagging strand. Watch this one. The lagging strand, you remember, we have to lay down the primer and then make the DNA. Oops. All right, here we go. Lay down the primer, then make the DNA. Lay down the primer, then make the DNA. Lay down the primer, then make the DNA. So, who lays down the primer? You tell me. Primates. Who makes the DNA?
DNA polymerase.
Which one? 3. Good. Excellent. Then DNA polymerase 3's cousin, DNA polymerase 1, says we don't need this primer anymore. So what does it do with it?
Take it out.
It removes it. And what does it replace it with? DNA, right? Watch this. Who seals the final gap? DNA ligase. Good. Did you catch all that? Let's do it again. What I'm going to do is extend this line so we can do it a couple more times. Okay?
Pink is the primer?
No, pink is the DNA now. The green is the primer. Remember, the leading strand goes all the way. But this one is made in pieces. So let's do it again. I have to make the primer. Then I copy the DNA. Then DNA polymerase 1 will come in. Okay, we don't need this primer anymore now. And it replaces it with DNA. Who seals the gap? DNA? Ligand. Are we catching that? Let's do it again. Who lays down the primer?
DNA polymerase.
No?
Wait,
Primates. Listen to the question. Who makes the DNA? DNA polymerase 3. Who removes the primer? DNA polymerase 1. Replaces it with what? DNA. Seals the gap? Ligase. You see how we're doing it? So it just keeps repeating. I can't do that last part. Do you see that? Your cell says, I can't do that. Because there's nothing to attach the nucleotide to. See, all this time when it had the primer, it had the attachment point. I don't have that anymore. What do you expect me to attach my DNA to? I don't have that. So look what happens. This is the original chromosome. What happens is it removes a piece of your chromosome. That's how it solves the problem. It says, I can't copy that, so let me remove that. So you do lose a piece of the chromosome every time you copy your DNA. Every time. So now watch this. Your cells are programmed to copy themselves around 50 times, no more. Because what really happens is it realizes that every time I copy the DNA, I'm losing a piece of the chromosome. That means the chromosome is shortening over time. So why is it set for the cell to replicate 50 times, and after that it cannot replicate anymore? Because this is what can happen. What's happening is on your DNA or chromosome, there are genes. We know that, hopefully by now. So let's say your DNA is copied once and you lose this piece. Okay, I'm fine, I'm okay. I didn't lose a gene. I lost a piece of the chromosome, but it's not a gene. Okay, I'm okay. I'm okay. I'm okay. I'm okay. I'm okay. Now now I'm bad. I just lost a gene. Okay, now I lost 2, and so forth. You're in trouble. So because every time you replicate the cell, you do lose a piece of chromosome, and genes are housed on chromosomes, you run the risk of losing a gene. Do you agree with me? Mm-hmm. So God, nature, whomever did it this way, designed it this way, it is amazing. The person who discovered this won the Nobel Prize, which is like $1 million prize. They're beautiful, right? And it said, nature, God, whomever did this, on our chromosomes, we're gonna put caps on our chromosomes. These are called telomeres. And the enzyme that puts the caps on is called telomerase. That's an enzyme that puts these caps on the end of our chromosomes. A lot of people equate them to your shoelace. You see that little plastic piece on your shoelace? It keeps your shoelace from fraying. The telomeres keep your chromosomes from shortening in the critical areas where there are genes. Now, they're still shortening. Listen carefully, please. Your chromosomes will shorten, and you cannot avoid that. But at least they're going to shorten in the telomeres. What are telomeres? They're repeat sequences. They are repeat sequences that mean nothing. They're buffer zones. They mean nothing. There are no genes in there. So, repeat sequence, repeat sequence, repeat sequence, repeat sequence, repeat sequence. How many of them? I don't know. It depends on mom and dad. Mom and dad tells you how many of them. Let's say you have 10,000 of them, I hope. That's good. Because every time you copy the DNA, you're going to lose the repeat sequences. But at least you're not losing the genes. It's a buffer zone. It knows this is going to happen. So now you're going to start losing telomeres, but not genes. So look what happens now, if we can diagram this in a better way, now that hopefully you're understanding this here. We're going to put the genes in relationship to the telomeres and see if we can make sense out of that. Here are your genes. These are valuable. You don't want to lose these. So God, nature, whomever puts telomeres at the end of your chromosome. The length of the telomeres is determined by mom and dad, your genetics. The interesting thing is, listen to it, telomerase is active only when you're developing as a child in most of your cells. That means you're born with a certain length of telomeres. Use them wisely, because that's all you're going to have. There are some cells in your body that keep the telomerase on. They're called stem cells. You have to be able to, those cells have to multiply for a long time, beyond 50 times. I'm saying most of your cells will turn this off. As soon as you're born, whatever telomere length that you have is what you have for most of your cells. So those cells start multiplying. You lose a telomere. That's okay. Multiply again. You lose a telomere. That's okay. Add another one. That's okay. Another one. Okay, I'm okay so far. I'm okay. Hang on. I'm losing most of my telomeres. I must have multiplied too many times. You see how it does it? It's like a signal to the cell. The only time you're going to lose a lot of telomeres is if you've already approached the upper limits. Then, once you get to a certain length of telomere, it programs the cell to age and die. Do not multiply again. It tells the cell, stop multiplying. Just age and die. There's a word for this. You're not responsible for it. It's called senescence. It tells the cell to enter senescence and eventually die. There's a reason behind that. It's fascinating. This stuff is no wonder why she won a Nobel Prize over this. We come back to DNA polymerase 3 to talk about this. DNA polymerase 3, when it goes to copy the DNA, it is very good at what it does. And what does it do? It makes DNA. But once in a while, it makes a mistake. Instead of T, it brings in G. That's a mistake. Nobody is perfect. It's because of how fast it does things. Look at what DNA polymerase 3 does. It reads the template, and it goes like this. AGC, GCC, GCT, 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 AGT. It's amazing how fast it does it. It's phenomenal. So it's expected that it makes a mistake. But the cool thing about DNA polymerase 3, it does this. Watch. AGC, GCT, GCT, mistake, GCT, GCT, GCT. Oh, I made a mistake. Break it down, fix it. And then go for it. I mean, amazing. You mean to tell me it proofreads its own mistakes? Yes, it does. We all say DNA polymerase 3 has proofreading capabilities. It is similar to you handing in a piece of paper to me for grading. But before you do it, you check for mistakes because you don't want to be penalized for it. The penalty of this mistake is potentially cancer. So it's saving you from cancer. The first place to fight cancer is right here. Once DNA polymerase 3 makes a mistake, it will tear the DNA down again to fix the mistake and then rebuild it. This is equivalent to you doing this. When you're writing a sentence, you misspell the word, for example, factor, for example. You misspell it. And then you keep writing. You look back and say, well, oh, I misspelled the word factor. But you're now already 8 words down the line, right? And then you take your eraser. You erase the 8 words to go back to the word factor, correct it, and then rewrite the words and keep going, you see? No, of course, we don't do that. We just fix the word factor there, right? But that's what DNA polymerase does. It tears it down to fix it and then rebuilds it again. It's phenomenal. But it does sometimes do this. GCT, 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 mistake, GCT, 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 then just keep going. That means you have a mistake. It made a mistake that it did not catch. Luckily, there are 2 other mechanisms that will check DNA polymerase III's work. And that's these guys. Mismatch repair. There's another mechanism called mismatch repair. It looks for complementary base pairing. A always goes with, what did you say? T. G always goes with C. What if your DNA polymerase III, by mistake, does this? A goes with G. That's a mistake that the mismatch repair will pick up. The mismatch repair will look at the sequence as A with T, good, G with C, everything is good. What is this? Can't have that. What are you doing? And change it. That's equivalent to you going to the word factor and changing the mistake. You think we only have one mechanism to back up this guy? No. We have 2 of them. The second one is called excision repair. Excision repair would do this. I'm going to draw it up here. Excision repair would be like this. You have a mistake right here in the DNA. Who made the mistake? DNA polymerase III, and it did not see it. Excision repair will come in there and say, break the DNA, break the DNA. Are you watching on the board? Erase this part and replace it with the good part. That would be equivalent to you doing this on the paper. Uh-oh, I misspelled the word factor. Take the whole paper, throw it out, and rewrite the whole thing all over again. That would be excision repair. Luckily, we have all 3 mechanisms. The best place to catch a mutation is right here at the time it made the mutation. But just in case DNA polymerase III oversees it, does not see it, overlooks it, mismatch repair got you covered. Excision repair will also help you. Now, if all 3 of them miss it, then you got a problem. If all 3 of them miss it, now you have a mutation right here. That's what this guy is built in for. Now listen, let me go back to the telomeres now. By the time your cell replicates 50 times, what's the chance of it having a mutation? 100%. You catch that? It's phenomenal what this guy is doing for you. It's saving you from cancer. It says this: by the time a telomere is so short, it means this cell probably replicated 50 times. That means this one most likely made a mistake that these guys did not catch. I have a mutation in a cell. I don't want it to replicate anymore because it's gonna develop a cancer. I'm gonna order it to age and die. It just saved you from cancer. It's amazing how that works. So the length of the telomeres, as it gets shorter, it's a signal that this cell is too old, and the old cell most likely has a genetic mutation. You don't want it to replicate, trust me. So what are people doing? When this was discovered, it was headline news back then, and they called it the fountain of youth, the telomeres and all that. Some people are thinking of, well, why don't we make it long, bring back the length so this cell survives? Do you really want that? Based on what I just said, you want this cell to survive and replicate? No, I don't want that. Yeah, because we got a problem here. The idea is when the length is short, that means it multiplied too many times, there's a chance most likely it has a mutation. If you genetically engineer, whatever, remake my telomeres, you're guaranteeing me long life with cancer. That's what you're guaranteeing me. You see, I don't want that. I want long life healthy. Okay, long life with cancer, that just extends my misery. I don't want that. So initially we thought, okay, extend the telomeres until we started realizing what these guys are doing and what they're protecting us from. And then there's another thing. When we look at cancer cells, these guys are amazing. Remember, when the telomere length is short, it signals the cell to stop multiplying. You're most likely a cancer cell. Guess who knows that? The cancer cell. And it will activate the telomerase gene. Remember, your telomerase gene is off. A cancer cell is a cancer because it reactivates the telomerase to force rebuilding your telomeres to keep multiplying. It's gonna fight you back. Your cell is saying, stop multiplying, you're a bad guy, you're gonna have to die. Your cancer cell, no, I'm gonna reactivate your telomerase to rebuild your telomeres so I can multiply forever because I'm a cancer cell. You can't kill me. It's amazing. See, they're called immortal. Cancer cells are referred to as being immortal. See, but doesn't really mean that you can't kill them. Of course, we can kill them with therapy and radiation and all that. It actually means I am immortal because I can multiply forever. I don't reach that 50 upper limit and I'm forced to age and die. I'm a cancer cell. I will reactivate your telomerase, rebuild your telomeres so I can break this rule and multiply forever. Is that amazing or what? We find that a lot of cancer cells reactivate the telomerase to rebuild the telomeres to keep the cancer cell multiplying forever. And that's why they're cancer cells. We talked about cancer cells losing contact inhibition. Remember, normal cells, when they touch each other, they stop multiplying. We said cancer cells don't do that. They lose contact inhibition. Normal cells, some cells have to attach to something for them to multiply. Cancer cells don't do that. But this one too. Normally, you lose your telomeres and the cell ages and dies. Cancer cell says, no, I don't want to die. I'm going to reactivate your gene, because I know you have the gene, it's just off. I'm going to reactivate it, rebuild your telomeres so I can multiply forever. Wow. It's amazing how that works. So when they talked about the telomeres being the fountain of youth, I'm going to rebuild your telomeres, and not my telomeres. My telomeres, I like them, what they're doing. I like the fact that when they get too short, they order the cell to die, because there's a chance that that's a cancer cell. Now the question is this. The length of the telomere that you're born with is what you're born with. So let's say you're born with a telomere this long. The idea is not how long the telomere is. It's more like how fast it shortens. That's more important. We know it's going to shorten. What determines how gracefully you age is the rate of the telomere shortening. If you go and meet someone on the street and they ask you, hey, how old are you? And you say, I'm 40. And they say, oh, wow, you don't look like a day over 30. Oh, you're beautiful. Like that. Your telomeres are going like this for you. See that? Because we found that telomere length determines the rate of aging to some extent. It also determines how soon you get sick and all those things. So if you're 40 and you look 30, you're aging gracefully because your telomeres are going like this for you. Nice. Now the other way. How old are you? 40. And they look at you. Ooh. You look 60. It's because your telomeres are going like this, like that. That's the difference. Okay? So the idea is not how long your telomeres are. The idea is how fast they're shortening on you. You can be biologically 50 years old. Sorry, chronologically 50 years old, biologically 70. So I'm 50, for example. I'm not 50, I'm more than that. But let's say I'm 50, but I'm chronologically 50, my birthday, whatever. But biologically, my telomeres are this short. Oh, I'm in trouble. Because I'm going to start getting sick maybe next year or so. You see? But if I'm 50 and my telomeres are this, oh, I got a long way to live there. See? It's all about the telomere length. That's when they said, well, let's make it longer. And then someone said, hang on, they're actually doing something here. I don't know if I want to make them longer then. They're actually a protective measure. The fact that they shorten on you is because there's a chance that you have a mutation there. So it's all about how fast they disappear on you. Now we already know through research that we can make the telomeres shorten slower based on our behavior, our day-to-day behavior. I mean, you can get a test to see what your chronological age is. So like a $250 test. Yeah, it's really easy. $250 test. You can send a sample in, and I don't know what they ask, probably a blood sample or something, or saliva maybe, I don't know. But they tell you what you are chronologically versus what you are biologically. And they're gonna tell you this, you want your telomeres to shorten slower? Yeah, everybody's gonna say yeah. Eat right, exercise, no smoking, no drinking. Well, I'm already telling you that, and it's for free. You see? You don't need a test to tell you, eat right, no smoking, no drinking, exercise, lead a healthy life. You don't need a test to tell you that. We all know that we need to do it. But the test confirms what you are chronologically versus what you are biologically. Sometimes it's bad news. Sometimes it's good news. So, is that pretty cool or what?
Yeah.
I love this telomere story. No wonder why she won a Nobel Prize.
That's interesting.
Good. I'm glad I got your attention. Go, take a 7-minute break, you guys. Yeah, 7 minutes. I'm counting the second. Got another chapter to go through. That's chapter 15 for you. I mean 16.